So welcome everybody to today's monthly webinar. As usual, we will have the speaker with talk, which will be around 30-40 minutes, and then there will be a QA session. So if you have any question for Irene, which is the speaker of today, you can wait to the end of the talk, or you can also write them in the chat. And with this, I will introduce Irene. So today, speaker is Irene Cobo Simón. She is a postdoc at the Institute of Forest Science in Madrid, Spain. So she is there giving the talk before dinner. Before that, she was at the University of Connecticut in Jill Werkrit's lab. And she led as a postdoc the development of the web-based application CartograPlant, which she's going to talk about in a few minutes. And she's interested in understanding the adaptive potential of forestry under a climate change context. And with this, Irene, if you want to share your screen, and you're ready to go.
Okay, thanks, Ana Rita. Okay, I'm sharing my screen right now. Okay, can you see my screen? Okay?
Yes, yes.
Oh, thank you. Okay, well, hi everyone. Thank you for being here today. As Ana Rita just said, I'm currently a postdoc at the Institute of Forest Science in Spain. And before that, I performed a 2-year postdoc at the University of Connecticut, where I work in the development of CartograPlant. So CartograPlant, first of all, is a web-based application that integrates, visualizes, and analyzes genotypic, phenotypic, and environmental data from georeferenced plants, including forest trees. And this web-based application is relevant and timely nowadays because, as we all know, climate change is threatening plant health and productivity at all spatial scales. However, it remains unknown whether plant breeding can keep pace with the rate and direction of this environmental change. On the other hand, the incidence of invasive pests and pathogens is increasing due to globalization, and this tendency is being exacerbated by current climate change. On the other hand, other specific threat to forest trees is the illegal logging and deforestation. So in order to deal with all these threats to plants in general and to trees or forest trees in particular, the match between the genotypes, the phenotypes, and the new environments will allow us to identify, for instance, candidate genes that can confer resilience to these biotic and abiotic stresses. And also, the timber origin can be identified by using or by integrating chemical, genetic, and anatomic tree data, which can help us tackle this illegal logging and deforestation. So as a result, the tools that collect, integrate, and facilitate all these data, such as CartograPlant, are critical nowadays. So as I have mentioned at the very first of the presentation, CartograPlant integrates, visualizes, and analyzes data, in particular genotypic, phenotypic, and environmental data coming from different studies. But the question is, how can these disparate data types that come from different studies can be integrated? And this is thanks to the metadata collection using standardized terms like ontologies and standards, that it is also accomplished by CartograPlant. This raw data and the associated metadata is available in the CartograPlant by 2 different sources. One of them is by thanks to the direct submission of this data via the Triple Plan PubGen or TPPS pipeline. And the other source of data is thanks to the biocuration efforts of our affiliated databases. Regarding the TPPS pipeline, this pipeline allows to store or to upload to CartograPlant genotypic, phenotypic, and environmental data, and also the associated metadata from a wide diversity of studies, including population genomics, association mapping, and landscape genomics studies. And it offers a specific DOI to each study. In addition, as I have mentioned many times during the presentation, the metadata is also collected using ontologies and standards, for instance, MIAP for the phenotypes. And these metadata collections ensure the so-called fairness of the data, which is its findability, accessibility, interoperability, and reusability. In addition, this TPPS pipeline supports the standard genotyping file formats such as BCF, which is very convenient for analyzing the data for data analysis, which is another feature offered by CartograPlant. It is also worth mentioning that the vast majority of the TPPS submitted studies are available in CartograPlant thanks to the efforts of our biocuration team. And regarding the environmental data, because I have talked about the genotypic and the phenotypic data in general coming from the studies, it is also available thanks to the environmental layers that are available in CartograPlant. Currently, we have a total of 950 environmental layers, but it is worth mentioning that these environmental layers are constantly growing. So because we are, our informaticians are constantly including new environmental layers in CartograPlant, and that allows to ask a wide diversity of biological questions. As a result of all this data integration, CartograPlant currently hosts about 8 million of plants belonging to 600 species, about 300 genera, 43 countries, and about 300 studies. So in total, CartograPlant hosts about 700 million of genotypes, 1.7 million of phenotypes, and as I have mentioned before, about 950 environmental layers. Regarding the data visualization, because again, CartograPlant integrates, visualizes, and also analyzes data. This data can be visualized thanks to a very user-friendly interface, as you can see. This interface is divided basically into two panels. The so-called left panel, which is the action panel, where the users can interact with the map and the plants data, and it is located in the left part of the screen. And the right panel, which is a worldwide interactive map, where that shows basically the plants and the environmental layers that has been selected on the left panel. And regarding the environmental layers, they can be visualized by using the left panel. So by clicking this button here that says environmental layers, users can display all the environmental layers available in CartograPlant. And once one of these layers is displayed, as I have mentioned before, the right panel offers a worldwide map that it is interactive. So by clicking directly on the map, anywhere on the map, you can display information about this specific point. For instance, as you can see, the latitude, longitude, and the elevation. And since in this case, I am displaying the major soil groups, you can also get information about the soil type and a brief description about this soil type. In addition, users can also display information about the plants located in the map. So by clicking directly in one of the plants located on the map, information about this plant, this specific plant, is also displayed. So again, the latitude, longitude, and the elevation information, also taxonomical information about the plant. And as you can see, there are another two buttons here. One of them that says Add Plant allows users to select this plant for further data analysis. And the other that says Study Info allows to display specific information about the study to which this plant belongs. So as you can see here, when you click at this button, you can see, for instance, the name of the study to which this plant belongs, the authors, the publication year, the year of publication, and also you can download the data files that are associated and available in CartograPlant to this study. In this case, you can download the phenotypes and the genotypes of the plants. And also you have information about the type of markers, in this case, microsatellites, and the number of markers available for this study. Even if this study shows the microsatellites, the majority of the studies provides SNPs, which are the most widely used molecular markers nowadays. And finally, you can select plans for further data analysis, and it can be done by two different ways mostly. One of these ways is by drawing directly on the map a polygon, thanks to this polygon button here. And the other way is by using the left panel. So by using the filters tab, users can filter the plants displayed on the worldwide map by different criteria, for instance, by family, genus, species, or mark the molecular markers, the study title, etc. And once the plants are selected for data analysis, authenticated users can click this Analyze button here in order to perform data analysis. So once you click at this Analyze button, this window is displayed. And as you can see, there are several tabs. So the first one that says Manage, well, here it provides a unique analysis ID for your analysis, and also you can create the analysis name and also select the type of analysis that you want to perform. In this case, it is selected genotype by phenotype by environmental data, but you can select any combinations of genotype, phenotype, and environmental data. And finally, the users are strongly encouraged to create a workspace. So by creating a new workspace, by clicking at this blue button, you can store the outputs of your analysis for this or for future analysis, which is very convenient. And once you have created your workspace, well, you click this next button here. And in the following tab, you can filter the phenotypic traits available in your selected plants. So in this case, the selected plants belong to two different studies, which is also displayed. And also this tab displays the overlapping status of the phenotypes. That is, for instance, which phenotypes are common between the two studies in this case, to which your selected plants belong, which is very useful for further analysis. For instance, if you want to do a meta-analysis, you need that the phenotypes overlap among the plants belonging to the both studies. And also, you can also display, once you select one of the phenotypes of interest, several histograms are displayed showing the distribution of your phenotypes throughout the individuals, the plants. And these histograms are interactive. So that allows you to select which part of the distribution you want to select for data analysis, depending on your biological questions. For instance, you may be interested in select the central part of the distribution or the tails, depending on the questions you may want to ask. And finally, if you select more than one phenotype, a PCA is displayed as well, showing the variance of your data as a means of data exploratory analysis, which is also very useful for to explore your data before doing any kind of data analysis and before selecting the phenotypes of interest. Once you have selected the phenotypes of interest for your analysis, that as you can see, they are displayed on the top of the page. You can see the analysis ID, the number of phenotypes, genotypes, and environmental layers selected. In this case, we have selected 3 phenotypes so far. The next tab allows to filter, in this case, by genotypes, by genotypic data. So again, the 2 studies to which your selected plants belong are displayed. And also the genotypes from each of the 2 studies are displayed in a Venn diagram that shows the overlapping status of the genotypes. So in this case, you can select, for instance, the SNPs, in this case, the SNPs that overlaps among the 2 studies, or those that are non-overlapping among the studies, depending on the questions you may want to ask. And we are also working in providing here a workflow or a way to remap the SNPs against the same version of the reference genome. Sometimes. I'm sorry.
Oh. Sorry.
I'm hearing a noise. Oops.
Sorry, Irene, go ahead.
Oh, no problem. Yeah, I thought that someone was asking something. Okay, so we are also working in providing a a way to remap the SNPs against other versions of the reference genome, because sometimes you don't find overlapping SNPs among two studies, not because they don't exist, but because the SNPs are mapped against or are obtained against different versions of the reference genome. So this way, the users can have on the one hand, the SNPs the most updated as possible, and on the other hand, you can make your SNPs comparable, make SNPs of different studies comparable, and this way you can do again meta-analysis, like an analysis using data coming from different studies. Once you have selected your genotypes of interest, then the following tab allows you to filter these SNPs by quality, so performing a quality filtering of the SNPs, and also the imputation of the lacking SNPs for further data analysis. So we provide several ways to perform this quality filtering, such as, for instance, several approaches. For instance, by using the missingness per individual or the minor allele count, etc. We provide the most commonly used criteria to filter by quality your SNPs, and also we provide the possibility to impute the lacking SNPs by using a program that is called LinkImputer. So this program allows to perform this imputation without the need of a high-quality reference genome. So it is also suitable for non-model species, because it is based in linkage disequilibrium between the SNPs. And also we are working in providing histograms to help users decide the best quality filtering thresholds for your quality filtering, your SNP quality filtering, because it depends highly on your data set, and sometimes it is tricky to find the best filtering threshold for your data set. So having a way to visualize your data and how the different thresholds will affect the quality of your data will be also very useful, but they are not available yet. And once you have filtered your SNPs, you can perform population structure in the following tab. Currently, population structure analysis. Currently, we provide only one approach to perform this population structure analysis, which is the program FastStructure. But we are also working in providing other approaches such as PCA or DAPC to perform this population structure analysis, which is another. It is interesting by itself, but it is another way to perform a data exploratory analysis before the further analysis. And the last step is the selection of the environmental data of interest for your analysis. So in this tab, you can see all the environmental layers available in CartograPlant. And if you select more than one, a scatter plot is automatically displayed, showing the collinearity or the collinearity, possible collinearity between the variables, which is also very useful in order to help you decide the most suitable environmental data for your data sets and to avoid kind of redundancy or selecting layers or environmental variables that are telling mostly the same story. And finally, the last tab allows users to perform data analysis. So in this tab, we currently have several analytical workflows available, but we are working in providing more very soon. So for instance, if you click on one of them, in this case, the genome-wide association analysis, you can see how a brief description of the workflow is shown, and also a kind of the input files that are needed for your analysis, that you can access them in your workspace. So among the data selected in the previous tabs, for instance, the SNP data or the phenotypic data that you may need for your data analysis. And this is a kind of draft showing the workflows that are currently available in CartograPlant that are highlighted in red, and the workflows that will be available soon. We are working on developing and to provide in CartograPlant. So this picture tries to show a little bit the logic behind the reason why we have selected all these specific workflows for CartograPlant. So again, CartograPlant provides phenotypic, genotypic, and environmental data and the associated metadata. And all the workflows are focused on making the most of the potential of CartograPlant. So for instance, we start in the case of the genotypic data. Of course, we provide a SNP quality filtering and the SNP imputation, as I have explained before. And we are also providing the possibility of map the SNPs against the same version of the reference genome, because the ultimate objective of CartograPlant is to allow mega or meta-analysis. So the main difference between these two approaches is that both of them perform analysis using data coming from different studies, but mega-analysis makes use of the raw data among studies when it is available, whereas meta-analysis uses the summary statistics of other analysis, such as, for instance, genome-wide association analysis or landscape genomics. And this is used when the raw data is not available. And in both cases, it is necessary the metadata from the studies, which is another strength of CartograPlant, because again, CartograPlant collects the metadata from the studies, which allows this both mega and meta-analysis. So once the SNPs are remapped against the same version of the reference genome, it is also possible to perform population structure, as I have mentioned, population structure analysis, which is necessary for these two kind of analysis that combines phenotype and genotype data, which is the genome-wide association analysis, or genotype and environmental data, which is the landscape genomics analysis. And finally, with the summary statistics of these two analyses, it is possible to perform meta-analysis in case the raw data is not available. And just as a conclusion, CartograPlant is basically intended to serve as a community resource for plant molecular ecology. And the flexible analytical workflows provided by CartograPlant will allow users to analyze a wide diversity of data types, and also a wide diversity of experimental designs. And as a result, we facilitate diversity of biological questions, to answer a diversity of biological questions. As I have mentioned in the previous slide, the ultimate objective of CartograPlant is to allow the performance of mega and meta-analysis. And it is possible thanks to the integration of these data types from different studies, thanks to this metadata collection. Another strength of CartograPlant is that it is continuously growing to stay up to date with the latest research and statistical methods to analyze this high throughput biological data. And finally, having this centralized and up-to-date platform that integrates, visualizes, and analyzes high-throughput biological data is key in the current big data era in plant biology, because currently the majority of these data are located in different platforms and in different locations and usually lack standardization. So the researchers spend a lot of time trying to get this data from different sources. And finally, we'd like to thank all the members of the project. Without their hard work, this project wouldn't be possible. And of course, to you all for your attention.
Thank you, Irene, for the great presentation. So I think now there is time for question. Okay, Leonor has one in the chat. She's asking, is there a report generated that goes with the analysis that shows all of the dataset, software version, and analysis parameters?
Well, the information about, or the outputs of the analysis are stored in the workspace. If that was your question, I don't know if I have answered your question, but the software version and everything, I don't think that we have this information in the workspace. We have it, of course, the developers and everybody in CartograPlant have it, but I think it's a very good suggestion that we can include in CartograPlant, like what are the versions of the programs that are being used at this moment. Because it can influence the outputs, of course, of the accuracy of the analysis.
So yeah, I was referring in terms of like sort of like a reproducibility report. So if you publish something based on that analysis, other people could reproduce it or know what all the parameters were.
Oh, yeah. The parameters, of course, yeah, because the users can select the parameters. For instance, the thresholds for the quality filtering, etc. You can select them. So you are always aware of what parameters are you using in your analysis.
Okay, Monica, she raised the hand. Monica, if you want.
Yeah, sure. Thanks. Nice presentation, Irene. I have a couple just sort of questions about how you do stuff and how the platform works, just as from like a database person to a database person. So yeah, this platform has a lot going on. So I was curious about the back end, and then how you actually manage to keep all the load all the content, since there's a lot of content in there, and keep it updated, with the metadata that you talked about. Do you have like a Galaxy backend for the analyses, and do you use Triple to store the genotypes and phenotypes?
Yeah. I forgot to mention the Galaxy part. Yes, yeah. Thank you for asking me that. The data analysis is possible thanks to a Galaxy instance. So the CartograPlant makes use of a Galaxy instance to perform this data analysis. And yeah, in the storage, we can say, of the data is performed thanks to Drupal. So yeah.
Okay. And the map metadata, is that also in Triple?
I'm not entirely sure of that. I will have to ask the developers of the database, but I think so, but I'm not 100% sure.
Okay, just curious, since there's lots of diverse data types, and where you put it. And my other question is, just since it's so much data, how many curators do you have, and how hard is it to train them?
Well, they change because they are normally undergraduate students. But yeah, we have a lot of people. We have normally like 5 people, something like this, but yeah, they are constantly changing, because some of them graduate and then another person start. And it's not that hard to train them because they have a biological background. So normally they understand the papers and everything. But yeah, for instance, the format of the data and everything, it's a little bit more tricky. It's just, but it's a huge work. They are like, they are doing a great job.
Thanks.
There is a question in the chat from Suk. Is there any way for users to add private data and do the analysis with their data and public data?
Yeah, we have there is a possibility to upload your own data from your laptop, for instance, to perform a data analysis, even if it is not available in CartograPlant. But it is not available in CartograPlant, so you have to ensure that the format is correct and everything to perform the data analysis. So
I just wanted to add that, so is it possible to combine the data, public data in the CartograPlant and their private data and do the meta-analysis together?
Yeah, I think so. At least I don't know if this possibility has been removed recently, but at first it was possible. So you can upload data from your laptop, for instance, and combine it with the CartograPlant. But the problem is that sometimes the format is not correct. So I think it is always advisable just to, if you want to include your data, maybe it is better to upload it to CartograPlant via TTPS, which is the pipeline that we have to upload data to CartograPlant. And this way you make sure that it is in the correct format, and then you can use it for your data analysis. I think that will be the best way to do that, because otherwise the format maybe is not compatible.
But then uploading means making it public, or is it a way to keep it private after loading?
Oh yeah, if you upload it via TPPS, it is public.
Yeah, okay.
Oh yeah.
Thank you.
I have a question regarding like the phenotypes because so as I understood, you are combining data phenotype, like phenotypic data across multiple studies that they are in CartograPlant, and like do you require that these phenotypic data have specific standards? I mean, like there can be some differences in how they have been collected in the different studies.
Yeah, this is another tricky part of CartograPlant. So yeah, this is why we use these standardized terms. Like for instance, for phenotypes, we use MIAPI, which allows to have the all the phenotypes in the same, with using the same standards. We can say it makes the phenotypes comparable between different studies, because there is this problem, like the different ways to measure the data. But thanks to these ontologies and standardized terms, we try to make it comparable.
Okay, so you use the ontologies to kind of map them. So yeah. There is also one comment in the chat from Marcela. She say, we have a standard for genetic variation working group in the AgBiodata Consortium. I will be interested in checking the SNP data you guys host. How many species, metadata? Maybe we could coordinate submission to the European variant archive.
Okay, that would be great.
Yeah, so maybe you can follow up with Marcela about that.
Yeah, sure. I think I have my, I think Ana Rita can provide you my contact.
Yeah, so Irene, she's also on Slack, right?
Yeah.
She's part of the genotype to phenotype working groups, but for sure I can put you in contact. Yeah.
Thank you.
I don't know if Sue raised the hand to
Yeah, I have a question. So this might have been brought up and I wasn't paying attention earlier. Sorry if that was, but I was wondering if someone has access to data that's not yet published, and can they upload just for their own analysis and then release it to the public after the paper's been submitted?
Yeah, I think I think that all the data that you upload via TPPS should be published, because it has to belong to a study. So you have to provide a title for your work and everything. So yeah, it has to be definitely published. But again, like I think Suk asked something similar before. So there was a way to, but I don't know if it is currently available. So I have to check it. But before you can, in previous, like, I don't remember when exactly, but it was possible to upload to CartograPlant in the data analysis tab. You can upload your own data and perform data analysis. But the problem is the format of the data. So if it is not in the correct format, probably you are not able to perform data analysis in CartograPlant. So again, the best way to perform a data analysis in CartograPlant is to upload first your data via TPPS, and for that, it has to be published, because it needs to be vinculated to a published study, and then use it for data analysis.
Can I ask a follow-up question then? So then can we download an instance of or can we download the web application and have our own local instance while we analyze the data internally?
So download the CartograPlant, you mean?
The software, yeah. Can we download the web application?
I don't know. I'm so sorry. I have to check it with the informaticians. I don't think that it is possible, but I cannot guarantee you that 100%. So I'm not sure. I'm not sure. I have to check it with the informaticians.
Okay, thanks.
Yes, I thought Adam had the raise your hands, but maybe.
Yeah, I had a similar question. I was in the same vein of thought of, like, do you know if it's running in AWS or on a cloud like that?
Sorry, where?
Like, where is the software running?
The software of CartograPlant, I'm not very sure about that. I was during my postdoc, I was focused on the workflow development, but the web page development was run by informaticians. So they can ask you this question better than me. I'm not entirely sure how it works internally. So sorry about that. I can check it with informaticians and go back to you.
Yeah, I was
I can
curious
speak
for
to
the
that.
same reason as Irene, like if it is possible to download it and run it. Right.
Yeah.
It is a triple extension module, so I think if you have triple system, you can download and run. I think because it's a triple extension module and they have GitHub repository and all that. So.
Yeah, I can see the code there. It would be the back end, I guess. Yeah, for the front end, it seems like you could have back end. It's okay. Yeah, thank you.
Yeah, it seems that Sean replied in the chat that it will be possible to host your own instance of CartograPlant, but there are quite a few components. So you already.
Yeah, so that is the long-term goal. As for right now, we were still, it's very in development, so we haven't published, a user guide for installing it. And like I said, it does have quite a few components. We have a separate GeoServer database that stores all the layers that you saw on the interactive map. And it does require, a triple site that stores all of the study data. And to answer the other question about where it's hosted, it's currently hosted in this instance, the TreeGenes and CartograPlant instances is hosted at the University of Connecticut on the cluster.
Thanks, Sean.
Thank you so much, Sean. I didn't know that you were here.
For answering the technical question.
Yeah, he's one of the informaticians that are developing CartograPlant, so the best person to ask your question.
Yeah, thank you for joining us and be of support to Irene. Are there any other questions for Irene?
Are you based in Connecticut, Irene?
Sorry?
Where are you based?
Oh,
Just
no,
curious.
right now I'm in Spain.
I missed the first. I missed your introduction. I'm so sorry.
Yeah.
Actually, I'm in Spain, but I performed before the current postdoc I'm doing right now in Spain. I performed a 2-year postdoc at the UConn, and this is where I work in the development of CartograPlant. And I still collaborate with them.
Any more question for Irene? If not, I will thank Irene for giving this wonderful presentation for us today, and thank all of you that join us for the webinar. And as always, I will post the recording on our website and our YouTube channel. So thank you so much, and I hope to see you again next month. Thank you.